Welcome back to another episode on our YouTube channel. Hello everyone, this is Hooi Leng here from Novel Bacteria and Drug Discovery Research Group under the Microbiome and Bioresource Research Strength at Monash University Malaysia. Today we'll be continuing our topic on SARS-CoV-2. Just to refresh our memory, this virus is an enveloped RNA virus. It presents crown-like spikes on the outer surface, which resulted in its naming as coronavirus. Recently, our group has published a study looking at 7 genomic sequences of SARS-CoV-2 virus isolated in Malaysia. The first case in Malaysia was reported on the 24th of January 2020, which involved a tourist. After that, the Malaysian government has announced movement control order to break the chain of transmission. The Ministry of Health has been working with several government agencies to set up contact tracing system so that they could identify and attend to suspected cases as soon as possible. So, how do we obtain genomic sequences of this virus? Frontline healthcare workers will interview patients and collect their swab samples either from their homes or hospitals after obtaining their consent for testing. Upon arriving at the laboratory, the lab scientists will then perform RNA extraction and real-time PCR. Depending on the presence of viral DNA, diagnosis will be made. In order to understand the virus, researchers will then perform whole genome sequencing of this virus and study their genomic composition, particularly to identify any changes that occur throughout time and geographical location. In response to call to accelerate data sharing, researchers are now submitting viral genome sequences to public databases. By doing that, it enables global collaboration and speeds up the understanding of this novel coronavirus. Following the new normal and work-from-home policy, our team is now joining the fight against COVID-19. We are the first research group to publish phylogenetic analysis of SARS-CoV-2 genomic sequences that have been isolated earlier of this year in Malaysia by comparing them to other strains devastated in public databases. Diving deep into their genomic codes, what we noticed was that most of these strains showed high similarities with strains isolated from Asia region. For instance, those in clade 3 and 4 were closely related to strains isolated from Singapore, Japan, and China. Furthermore, there are numerous differences across genomic regions encoding for structural proteins including spike and membrane proteins. Bear in mind that these features are important points to consider when it comes to vaccine design. Along with that, researchers are now observing a highly infectious strain with a nucleotide change known as D614G strain. Even though it is quite normal for coronavirus to mutate over time, the emergence of such strain with high transmission rate needs to be studied thoroughly to prevent further spread of COVID-19. On the 10th of August 2020, the Malaysian Ministry of Health has reported potential super spreader in one of the clusters, and within a week, it was then confirmed that 4 cases were carrying this mutation. Nonetheless, due to the swift actions by the health authorities, these clusters are now under control. The fight against COVID-19 is still not over yet, but together we can beat this pandemic. Always remember to practice preventive measures including social distancing and self-hygiene practice. Thank you for watching and stay tuned for upcoming videos. Don't forget to click like and subscribe button on our channel. See you!